Opportunity to welcome you back to this other session of ours. So this is one of the lesson which summarizes autotrophic nutrition, and we are going to look at factors which affect photosynthesis. We've been studying this from P4. So one is carbon dioxide concentration. We shall talk about it. Light intensity, temperature, chlorophyll concentration, oxygen concentration, water and dissolved nutrients, enzyme inhibitors, e.g. the cyanide, the DCMU, which is dichlorophenyl dimethylurea, that is DCMU. Another one, we have some air pollution or some air pollutants, e.g. the sulfur dioxide, the altitude, and lastly salinity. So when we talk about salinity, salinity is mainly salt, or the concentration of what? Salt is what that is what salinity means. Then from there we shall go ahead and look at the principle of limiting factors. So it states that at any given moment the rate of a chemical process is limited by the one factor which is nearest its minimum value, and by that factor alone. So that is how the principle of limiting factor states. So for more information about this, just go and open the biological science has got the clear information, or more clear information about what? The principles of limiting factors. So let us proceed. We're going to look at the graph illustrating the concept of limiting factors on the rate of photosynthesis. We say that the limiting factor is that factor at any given moment the rate of a chemical process is limited by the one factor which is nearest its minimum value, and by that factor alone. So when you look at this graph below, yeah, that one, we have light intensity. We have light intensity which is down here. We have light intensity which is down here, the one which is there. Then you have the rate of photosynthesis, that is there. Now when you look this side, this first part here, we are seeing that the photosynthesis is limited by, carbon dioxide. Here photosynthesis is limited by carbon dioxide. In other words, carbon dioxide is acting as the limiting factor of what? Photosynthesis. So when you move to this part here, or this stage, we are seeing that photosynthesis is limited by what? By temperature, where we are having low concentration of carbon dioxide and low temperature. We are saying that photosynthesis is limited by temperature. Then when we move to this part where we are having photosynthesis is limited by some other factors, here we are having the high concentration of carbon dioxide, low concentration of what? High concentration of carbon dioxide, then low temperature. And up here we are having high concentration of carbon dioxide, high temperature. So when this condition reaches here, we are seeing that photosynthesis is limited by some other factors. So I want you to go and open that graph in biological science and read more about the principles of limiting factors and how they work. So the other part, let's look at salinity, and we are saying one of the major effects of salinity is osmotic stress, and hence there are intermittent relationship to drought stress or water loss. So this results in stomata closure in an effort to avoid the desiccation. So when we talk about desiccation means drying up in simple terms, with the reduced photosynthesis because uptake of with the reduced photosynthesis because uptake of carbon dioxide does what? Reduces. Then from there I want us to look at this graph carefully. Yeah, that is the graph I want to look at. So people have been seeing these graphs of autotrophic nutrition. Now, when we look at that graph, yeah, sorry for that inconvenience, but allow me proceed. Yeah, I was going to talk about the effect of carbon dioxide in the atmosphere. So we are saying that in the atmosphere, the concentration of carbon dioxide ranges from 0.03 to 0.04%. So that's the concentration of carbon dioxide which is in the what? In the atmosphere. We are saying that it varies between 0.03 to 0.04%. Now when we look at that graph, we are seeing that this side we are having, down there we have temperature, and this side up we have what we are calling the photosynthetic rate, that is the PS rate in microm carbon dioxide concentration in meters per second. Then up here we are having C4 or C3 plants at high carbon dioxide here. C4 or C3 plants at high carbon dioxide. Then down here we have C3 at low carbon dioxide concentration. Then from there let us proceed. I just want you to listen and be observing. So in one of the observation, you are seeing that generally the rate of photosynthesis increases gradually with the increasing carbon dioxide concentration to a maximum in C3 plants and in C4 plants. The explanation for that is that the rubisco carboxylase attaches carbon dioxide instead of oxygen because the carbon dioxide concentration is higher than the oxygen what? The oxygen concentration. Or you can say that more cells photosynthesize because of the increased carbon dioxide molecules available. Then the second observation we can look at from that graph of ours is that the rate of photosynthesis is faster in C4 than C3 what? C3 plant. So the explanation for that is that the PEP-C or the PEP carboxylase enzyme of C4 has a higher affinity for carbon dioxide than the rubisco carboxylase. So this rubisco carboxylase, for it has a lower affinity for carbon dioxide, and it is found in C3 plants, hence acts what? Acts faster. Now the other explanation for the other observation, I've so far looked at two observations. The third one is that the overall photosynthetic products are greater in C3 than in C4. We are saying that the overall photosynthetic products are greater in C3 than in C4 what? In C4 plants. The explanation for that is that C4 needs more energy than C3, which generally reduces the photosynthetic output. I hope you have got that. So another observation we can identify from that graph is that C3 plants have a higher compensation point than C4 plants. So I'm going to talk about compensation point in the next part after looking at some of these factors. So the explanation for that is that the PEP, this is because the PEP-C has a higher affinity for what? For carbon dioxide. Then another observation we can observe from that graph is that the C4 plants are more efficient at low carbon dioxide concentration, while the C3 plants are more efficient at high carbon dioxide concentration. Reason being that at low carbon dioxide concentration in C3, photorespiration reduces the photosynthetic efficiency, yet the PEP-C or the PEP carboxylase has a higher affinity for oxygen. So what I've been taking you through these observations and their explanations, the way you can see them there. So now what you do, you can pause this video a bit. You write those observations, talk the descriptions and the explanation. So allow me end here and sign out. I have completed looking at the effect of carbon dioxide. Carbon dioxide as the limiting factor. Now we are going to look at chlorophyll concentration. Is going to be our next lesson. Allow me wish you the best. I sign out. Please like, comment and share.